it's my pleasure to introduce Dr. Georgia Schafer. Dr. Schafer got her PhD from the Humboldt University in Berlin in Germany and performed her postdoc work here at the University of Cape Town in South Africa. She's currently leading the research group of virology emerging viruses at the ICGEB Cape Town component here in South Africa, where she investigates the entry mechanism for Kaposi sarcoma herpesviruses and the human papillomaviruses. In response to the current COVID-19 outbreak, Georgia's group has also set up several SARS-CoV diagnostics and pseudovirus neutralization assays to support various nationwide research activities. Georgia, again, it's a pleasure to have you here with us, and the floor is yours.
Thanks so much, Luis, for the kind introduction. I just quickly share my screen. All right, thanks again for this kind introduction, Luis, and good afternoon to everybody. As Luis said, I'm based at the ICGEB Cape Town, where I'm heading the newly founded virology group. And since I'm the first speaker this afternoon, I thought I'll give a very general overview of the importance of vaccines in controlling infectious diseases outbreaks. And I will also talk about different types and platform of vaccines, and of course, where the current COVID vaccines would fit in. And obviously, I will also touch on the current situation in South Africa, talk about the COVID vaccine efficacies and all these issues associated with that, and also the dependence of the country of sourcing COVID vaccines from abroad. And I will end my presentation with some thoughts for discussion where South Africa could move from here. So you are all aware that vaccination is the single most effective intervention against infectious diseases and in controlling pandemics. And this slide just illustrates that just by giving examples of historical mass vaccination programs in the US carried out against, for example, diphtheria, polio, or measles. And these vaccination programs have almost eradicated these diseases from the population. Now, currently, we are facing the COVID-19 pandemic with over 100 million cases to date globally. And you can see that 3.7 million of these cases have been reported in Africa, half of them in South Africa. And a similar picture can be drawn for the death rate. Worldwide, there have been over 2 million deaths to date reported, 100,000 of them on the African continent, again, half of them in South Africa. But there's obviously a great need for vaccination to control the pandemic worldwide. And as you know, a global hunt for a COVID-19 vaccine has begun just about a year ago. And in this short time span, we now already have a number of emergency authorized vaccines, which is enormous and historically unheard success in vaccinology. This is also illustrated here. Now, this chart shows you a timeline from the year a pathogenic agent was first linked to a disease to the year in which a vaccination against that pathogen was licensed in the US. And you can see that some of the vaccines took a long time to develop, and some of the diseases have not even an effective vaccine yet. And some of the vaccines did not take that long. So, for example, measles was probably the quickest vaccine ever developed. It only took 10 years. But this record was broken in 2020, and several vaccines were developed within just 1 year to fight the COVID-19 pandemic. And this was way faster than ever before, and importantly, without compromising on testing and safety. Well, this slide shows you a very simplified journey of a vaccine. It all starts with the development of various vaccine candidates and their testing for safety and efficacy in clinical trials. So most vaccines works on the principle that an antigen specific to the pathogen of interest, and then often in the presence of adjuvants, stabilizers, preservatives, etc., are injected to trigger an immune response against that vaccinated pathogen. I will talk about these different vaccine types and platforms a little bit later. So first of all, the vaccine candidate would pass through preclinical trials in the laboratory by in vitro and in vivo assays, and then it has to pass through 3 clinical trial phases, which I'm also going to talk about a little bit later. Only then it can be licensed and distributed. So when the results of these clinical trial phases are available, there are a series of steps that is required, including reviews of efficacy and safety for regulatory and public health policy approvals. And a vaccine must be proven to be safe and effective across a broad population before it will be approved and introduced into a national immunization program. So it's very important to highlight that the bar for vaccine safety and efficacy is extremely high, recognizing that vaccines are given to people who are otherwise healthy and specifically free from the illness. So once all these boxes are ticked, the vaccine can then be manufactured, not necessarily in the same country of destination, as I'm going to talk about a little bit later. And then the vaccine will be filled and packaged and transported to its destination. Now, depending on the type or platform of the vaccine, a cold chain might be necessary, and that can complicate things, especially when very low temperatures are required, which I'm also going to talk about a little bit later. So once the vaccine has been filled and packaged and transported to its vaccination, the vaccination campaign can start. And during those vaccination campaigns, further safety and efficacy monitoring takes place in an ongoing way. And this is also called post-rollout surveillance, which is very important. And these data are used to adjust the policies for vaccine use to optimize their impact. And they also allow the vaccine to be safely tracked throughout its use. Now, let me talk a little bit about the types of vaccines. There are three main approaches to designing a vaccine, and their differences lie in whether they use a whole virus or bacterium, just parts of the pathogen that triggers the immune system, or just the genetic material that provides the instructions for making specific virus or pathogen-related proteins. So the whole genome approach, the whole microbe approach can be divided into inactivated vaccine, live attenuated vaccine, or viral vector vaccine. So the inactivated vaccine refers to when the pathogen is inactivated or killed by chemicals, heat, or radiation. And this is the way, for example, the flu and polio vaccines are made. And these vaccines can be manufactured on a reasonable scale. And the live attenuated vaccines would use a live but weakened version of the virus, or one that is very similar to the virus or the pathogen. So for example, the measles, mumps, rubella vaccine or the chickenpox vaccine are examples of this type of virus. The disadvantage is that these vaccines cannot be used for people with compromised immune systems. And viral vector vaccines is a rather new approach of vaccines. Here, a safe virus is used as a delivery vehicle, and it carries the information for the expression of certain proteins against the pathogen of interest, which then can trigger an immune response and the actual virus vehicle, like an adenovirus, for example, would not be causing a disease because it's engineered in a way that it cannot replicate inside the cell. And the first licensed vaccine that used a viral vector approach was the Ebola vaccine. So that's a rather new development. And the subunit approach refers to vaccines that use only very specific subunits of the pathogen of interest, such as proteins or sugars. And the immune system would then recognize those pathogen-derived proteins or sugars and would elicit an immune response. So it does not contain the whole microbe or uses a safe virus as a vector. Most vaccines on the charted schedule are subunit vaccines, for example, the tetanus, diphtheria, or meningococcal meningitis vaccines, but also the HPV vaccine is a subunit approach. And then very new on the list is the genetic approach, which uses nucleic acids such as RNA or DNA for the expression of specific pathogen-related proteins that we want our immune system to recognize. And before the COVID-19 vaccine approach or hunt for COVID-19 vaccines, no such mRNA vaccines were available, and the COVID-19 vaccines are the first to be authorized for emergency use. Since particularly RNA is very fragile, these vaccines or these RNAs are wrapped into lipid nanoparticles and are stored at very low temperatures. And I'm going to come back to that a little bit later, but that causes some problematic issues, especially in low resource settings where a low temperature cold chain cannot be maintained. So what do we want our immune system to recognize? And I'll use the SARS-CoV-2 virus as an example, as all current COVID-19 leading vaccine approaches target the spike protein. Well, the spike protein, as you all know, stick out of the surface of the SARS-CoV-2 virus. It's a virus surface protein and the first seen by the immune system when the virus gets or infects a person. And this is also the protein that attaches to the human ACE2 entry receptor to gain entry into cells. So both during natural infection as well as during vaccination, which, as I said, uses spike approaches for immune responses. In both cases, neutralizing antibodies would be elicited by the immune system against spike, which would then prevent entry of new incoming virus via binding to the spike protein, as well as T cell responses would also be triggered in order to recognize and kill cells that are already infected and present spike or parts of the spike protein at the surface. So when I go back to the slide I just showed you before, I just wanted to illustrate the different current COVID-19 vaccine approaches, and it shows that there are many different platforms that are currently being used. And again, the common theme is that all these different platforms in one way or another use the spike protein for the body to elicit an immune response. So as I said, the mRNA approach is very new. And indeed, the COVID vaccines are the first mRNA vaccines that have been licensed. Again, the disadvantage is that they have to be stored and transported at very low temperatures. And then the Novavax vaccine is a subunit approach where the spike protein is being expressed and harvested and assembled in nanoparticles, and in the presence of adjuvants is being injected into the person and elicits an immune response against the spike protein. And a number of companies like AstraZeneca, Gamaleya, or Johnson & Johnson use the viral vector approach using an engineered adenovirus that is used as a vehicle to deliver spike DNA into the vaccinated person. And the DNA is then being used to express spike, and the body would respond with a response against the spike protein. Advantage is that the DNA is not as fragile as the RNA, which is contained in the mRNA vaccines, and also the adenovirus tough protein code helps to protect the genetic material inside. And then there are also some inactivated vaccine approaches, for example, by Sinopharm. So now I'll show you a very simplified scheme how the COVID-19 or in general vaccines will work. But again, it's extremely simplified. So let's say the vaccine, be it as an mRNA vaccine, a subunit approach, adenovirus or inactivated virus is being used to vaccinate people. It would enter cells and then start to produce the S protein in the context of mRNA or the adenovirus vaccines, or it would present the S protein at the cell surface in the case of like live inactivated vaccines or the subunit vaccine. So in any case, it will prompt the immune system to respond by producing antibodies via a B cell response against the S protein, and also by activating killer T cells to destroy potentially infected cells. That's exactly what would happen if the body encounters a natural SARS-CoV-2 infection. There would be antibodies that would be able to bind to the spike presented at the surface of the virus and would prevent entry via the ACE2 receptor. And there would also be activated T cells that would be triggered to recognize and fight the virus. Now, the hunt for a COVID-19 vaccine began in early 2020, and the exceptionally rapid progress seen last year also reflects years of efforts in researching and developing new vaccine platforms. As I mentioned, the mRNA vaccine was indeed the very first to be licensed in the context of the COVID-19 vaccine hunt, and also the first one ever to be licensed as an mRNA vaccine. And as I also mentioned, it is very important to emphasize that all vaccines went through clinical trials, were tested properly, and were proved to be safe. So, and also now additional monitoring of safety is ongoing as the vaccines get rolled out to more people following approval by the regulator. So safety monitoring will remain a key component of the vaccine rollout. So just to quickly illustrate the different phases of clinical trials, as I said, preclinical trials will be conducted in a laboratory by using in vitro and in vivo approaches. And then promising vaccine candidates would enter the clinical trial phases. In phase 1, the vaccine is given only to a small number of volunteers to assess its safety, to confirm it generates an immune response, to determine the right dosage. And generally in this phase, vaccines are tested in young, healthy adult volunteers. Then when the vaccine makes it to phase 2, it will then be given to several hundred volunteers to further assess its safety and ability to generate an immune response. And the participants in this phase have the same characteristics such as age or sex as the people for whom the vaccine is eventually intended. And when the vaccine passes phase 2, then eventually it would enter phase 3, where the vaccine is given to thousands of volunteers and is also compared to a similar sized placebo group. And most of these phase 3 trials are conducted across multiple countries in large international trials and at multiple sites within a country to assure that the findings of the vaccine performance apply to many different populations. So currently there are a handful of vaccines that have been approved for emergency use, and the list is constantly growing. And I just want to give you an overview of the leading COVID-19 vaccines that are particularly important in the South African context. As I mentioned, and I also showed you before, there are a number of companies that developed vaccines using different platforms. So we have 2 companies that developed mRNA vaccines. A number of companies used adenoviral vector approaches for viral vector delivery of the vaccine, or we also have here a subunit approach. And as I also said, all of these different kinds of vaccines in one way or another use the S protein to elicit an immune response against the spike protein of SARS-CoV-2. The schedule is also rather similar. Most of the vaccines need 2 shots, apart from the Johnson & Johnson vaccine, which only needs a single dose, which is very advantageous, especially in low resource settings. And the storage is also quite different between the vaccines. And again, the mRNA is quite a fragile molecule and requires storage at very low temperature, which is a disadvantage, again, in low resource settings. Other vaccines that can be stored at fridge temperatures are obviously much more advantageous. And also all these vaccines went through rigorous clinical trial phases. A couple of them have also been conducted in South Africa. And the first vaccine that received approval, as I said, was an mRNA vaccine developed by Pfizer-BioNTech. It received FDA approval in December 2020. Only a week later, the other mRNA vaccine developed by Moderna received emergency use authorization. And that was followed by the AstraZeneca viral vector vaccine in late December 2020. When you look at the efficacy, some vaccines perform better than others. Particularly the mRNA vaccines have an excellent efficacy, and these are data from these big clinical multinational trials. What is important to note is that authorities like the WHO or FDA say in an emergency situation, 50% efficacy is acceptable, and it is true for all the different vaccines that are listed here. Now, obviously, vaccinations have started globally, but there's also a huge difference between the different countries and the different vaccination rollouts. So to date, 150 million vaccine doses have been administered worldwide. That is equal to 2 doses for every 100 people. But again, there's a stark gap between vaccination programs in different countries, and only about 0.2% of all vaccinations have been conducted in Africa, on the African continent. You can see it looks rather gray, and this is also illustrated here. North America is quite good with its vaccination programs, but Africa is really lagging behind. So that is definitely a huge issue and reason to be concerned. So what were the plans in South Africa? So as I said, South Africa took part in several different vaccine trials, and the government, although rather late, accessed vaccines through COVAX and also via self-purchasing. And recently, about 2 weeks ago, 1 million AstraZeneca vaccines purchased from the Serum Institute of India have been delivered to South Africa, and rollout was planned to start in mid-February 2020 and first of all given to healthcare workers. However, there was a spanner in the works because of the emergence of the 501YV2 variant in South Africa. That's a variant that occurred first in October, November or so last year, is characterized by a number of mutations and deletions which confer selective advantage to the virus due to increased transmissibility and or immune escape, and is now the dominating circulating variant in South Africa, but also it spread globally already. And obviously this causes great concern for vaccine effectiveness. Now, the clinical trials that I showed you before were done globally, and also the numbers that I showed you were just numbers from global vaccine trials. And we only have bits and pieces of information how these vaccines perform in the presence of the 501YV2 variant. So what I showed you here, or what I'm showing you here, are those data that are available. We have some data on clinical trial, but we also have some neutralization data from the laboratory looking at vaccinated patients' serum, whether it still recognizes the 501YV2 variant. And you can see in these neutralization assays, there is a significantly reduced recognition and neutralization capability of patient serum towards the new variant. Then we look at these different clinical trials. Also, efficacy is quite reduced, and also depends on which endpoint is being looked at. So the AstraZeneca vaccine, which is the one that has arrived and was the one that was planned to be used for the first rollout, showed only a 22% clinical efficacy against mild and moderate COVID-19 disease. And because of this very large or largely reduced efficacy, rollout is on hold, awaiting clinical data on hospitalization and severe disease, because currently one does not know whether the vaccine protects against severe disease and death. And as Claudina just said, the Johnson & Johnson vaccine has arrived in the country. It shows a much higher efficacy, particularly when using the endpoint severe disease and death. So that is quite a promising endpoint data or result. And also the advantage of this vaccine is that it only needs one shot. So there's hope that the Johnson & Johnson vaccine will ease our situation in South Africa. And again, maybe the AstraZeneca vaccine will show that it can also protect against severe disease and death, because that's ultimately what we want, to reduce the severity of the COVID-19 disease. Okay, with that, I would like to end this presentation and just present you with a few thoughts for discussion. One is that South Africa, as I said, had to purchase its vaccines, the AstraZeneca vaccine, from the Indian Serum Institute of India. So there is no local manufacturing capacity of these large quantities of vaccines that are required in a pandemic like the COVID-19 pandemic currently. So we are dependent on import, which is something which is definitely an issue. Then we also need to secure fair pricing agreements. And as I said, South Africa took part in a number of clinical trials. And when we look at the AstraZeneca vaccine, for example, and South Africa took part in the AstraZeneca vaccine trial, in the end, it procured its first delivery of the vaccine from the Serum Institute of India for a price that was double the price of what European countries paid for the AstraZeneca vaccine. And this is something that could also be questioned. If communities take part in research and in clinical trials, there should also be some benefit and fair pricing agreement. And now we are even sitting with the AstraZeneca dilemma that it only shows a very low efficacy against our dominant variant in South Africa. The question is, can we continue using this particular vaccine now we have purchased it and collect data on disease severity, as I just said, because the ultimate endpoint is severe disease and death. And also, although the lab data show that neutralization is impaired in the presence of these of the new variant, and all the vaccines show a lower neutralization capability, neutralization is not everything, and also T-cell immunity plays an important role. So maybe this is also something one should look at in more detail and see whether these different vaccines do elicit a good T-cell and strong T-cell immunity, even in the presence of these variants, and can still be used against severe disease outcome. So what is the way forward? Definitely we have to acknowledge that the SARS-CoV-2 virus evolves. There are variants and they are probably occurring all the time. And genomic surveillance is very important to recognize and acknowledge these variants very early. And on that note, manufacturers need to adjust their vaccine approaches to accommodate SARS-CoV-2 viral evolution. So probably similar to seasonal influenza virus, and this is probably also something that will be ongoing in the future. But we should also use what we have, and this is the different vaccine platforms, and based on that, we can also start to adapt, license, and emergency approved vaccines. So maybe one can start giving 501(b)(2) based boosters. So it's basically a heterologous boost scheme. Also, one could look at the dosing regimen. Most vaccines are based on 2 doses, particularly in low resource settings. 1 dose would be obviously optimal for logistical and also financial reasons. And then I also think one need to recalibrate on how we respond to the COVID-19 pandemic and what we expect of the COVID-19 vaccines. So do we want vaccines that protect against infection, or do we rather want vaccines that protect against severe disease and death? And with these thoughts, I would like to end this presentation, and I thank you all for your attention.
Thank you, Georgia. Yeah, food for thought, but food for indigestion, really, because it looks like very tough questions that you've put out there. And so we can allow for one quick question, and then we'll come back for discussion later on, if there's anybody with a quick question. But I just wanted to say, the thing that worries me is that there's so much, it's so complex, there's so much virology here. And for healthcare workers, do they actually understand these things to be able to explain to recipients? Because that's why you've got all this vaccine hesitancy, because even healthcare workers themselves don't understand it. And I think what we're finding is they are themselves quite reluctant to take the vaccination. So how can we simplify the message, in other words?
Yeah, I guess there's also a lot of misinformation circulating on social media, etc., and that all contributes to vaccine hesitancy. But also the fact that the AstraZeneca vaccine doesn't show high efficacy or very disappointing efficacy does not really help in the whole vaccine hesitancy. And also the country itself did not respond very quickly in terms of ordering vaccines. So the first 1 million doses of the AstraZeneca vaccine is a very small amount given the size of the population in South Africa. So I think the signals that the government is sending are not exactly helping to overcome this vaccine hesitancy.
Yeah. And it's a global issue, but there's a question here from Swasti. So she says, does South Africa have the capability to develop its own vaccine?
Yes and no. So in South Africa, do you mean research or the manufacturing process? So that's 2 different questions. So we have the BioVac vaccine, which is half governmental, half research institution based, and they do produce vaccines, but on a much smaller scale than what is needed in a pandemic like this. So now we need large doses of vaccines quickly, and this is something South Africa cannot accommodate at the moment. There's the Aspen pharmaceutical plant, or I think there are 4 plants or so in South Africa that soon going to start packaging and filling the vaccine. So that is definitely a step in the right direction. But then there's also a number of research activities going on that look into developing new vaccines, not necessarily against the spike protein, because that's the one that mutates probably fastest and easiest, but also looking at vaccines that might target other SARS-CoV-2 proteins, such as the nucleocapsid protein, etc., which would only be expressed once the virus is inside. So it's something that the immune response would only recognize once a cell is infected, and then would start to, via cell-mediated immunity, try to kill those infected cells. So it's a different vaccine approach.
Yeah. And I guess it's important to say there's a difference between research and developing and then actually deploying the vaccine.
Yeah,
so I mean,
many steps.
a lot of us here are good vaccine researchers, but then deploying that vaccine is now the function of pharmaceutical development rather than benchtop. Louise, do you want to say something there?
I'd like to just thank Georgia for this nice vaccine. I'd just like also if Glodina has any comments about this vaccination in South Africa, because Glodina, as I said, is Director of Health Innovation at the Department of Science Innovation, but she's also part of the Ministerial Advisory Committee for Vaccines for the President, and then she can also make some comments if she wants. Especially in the AstraZeneca. Just to keep in mind, the AstraZeneca issue is for the South Africa variant that was mainly 90% of the sequences found here in the country. But this vaccine is currently utilized in other countries around the globe because it's effective for the strains, the variants that are there. But just a comment that I want to see, but if Glodina wants to say anything about vaccines.
Yeah, so she can make the comment now or later. Glodina, you want to make it now or later?
I prefer not to make any comments about the AstraZeneca vaccine. It is unfortunately that it landed before we could actually have the final results on the variant, but the variant sort of caught us all by surprise. And all I can say is that it's a celebration of the science, because we could immediately set up the system to actually develop a live virus testing facility for all vaccines that's going to be used now for all vaccines, and then also a pseudovirus that was developed, so that all vaccines will be tested against that before we actually go in further to test, to see whether it's actually applicable to South Africa or not.